Yes, I was a professor. I'm now emeritus professor at San Diego State University, and I have a position at Stanford University. But I still do some work with something called the bioactive lipids. The work that I have done for the last 30 or 40 years actually was inspired by an experiment I did on my second sabbatical that I did in 1990, when I was actually working with Romeo Betto and Giovanni Salviati and colleagues in the Salviati laboratory. And there was an observation that I'm going to tell you about that stimulated a complete change in my career. And as a consequence of that serendipitous discovery that was in the Salviati laboratory, I ended up subsequently founding two biotechnology companies from San Diego area. One is called LPath, and became a public company in 2005. And the second one was a company called MPEX, now called Vaxion. It's a privately held company, still in existence. Anyway, you probably heard of the proteome, the genome, but you might not have heard of something called the lipidome. And this is a pretty new concept. The concept of the lipidome is that there are a class of lipids that are signaling molecules. They're not lipids that make up, the energy sources. They're not the lipids that form the biological membranes that define cells, but these are a kind of a small lipids that we now understand are bioactive lipids. That is, they are signaled through receptors and ion channels, much in the same way as protein growth factors signal cells to perform certain functions. And the lipidome is very large. It's larger than the proteome. It's about 500,000 lipids large. And we suspect that there are probably probably at least 1,000, maybe 2,000 bioactive lipids that are good targets for drug discovery. That if you were to, that these lipids, growth factors can be dysregulated in certain pathological conditions, and if you can neutralize those bioactive lipids and prevent them from doing their signaling, then that could be a good target for drug discovery. And here are a couple of lipids that I've been working with for the last 20 years. One is called sphingosine 1-phosphate. You see, it's not a protein. It's not a nucleic acid. Basically, it's a single lipid. And here's another one, is a lysophosphatidic acid. It's derived from the glycerol phospholipid. And these are signaling molecules in the sense that sphingosine 1-phosphate or lysophosphatidic acid can act upon membrane-bound GPCR receptors or ion channels or other kinds of targets. And much like, as I said, a protein growth factor, these lipid signaling molecules can activate those receptors, and through the activation of the classical G-protein signaling system can modulate the behavior of the cell. They can ensure cell survival, promote migration, proliferation of cells, and are involved in many good processes like wound healing, but also when these signaling lipids get dysregulated and too much of those signaling lipids get produced, there's a problem, and that's a pathological condition, just like protein growth factors. If you have too much TNF-alpha, you get heart failure. So a little bit of TNF-alpha growth factor is a good thing. Too much of TNF-alpha is a bad thing. It causes inflammatory disease and even can promote heart failure. So these signaling lipids have a normal function, but if they get dysregulated, they can have a role in pathology. And so that's just what I said here. And what we discovered in my university at San Diego State University and the companies that this company called LPath that I founded, is that we could make antibodies in the laboratory against those signaling lipids. And by binding those signaling lipids with the antibody, we could prevent them the lipids from interacting with their receptors, and we could stop the signaling or mitigate the signaling that would otherwise be dysfunctional. And we developed those antibodies, an antibody to sphingosine 1-phosphate, we called it sonepcizumab, and an antibody to a lysophosphatidic acid, we call lapatamab. And those molecules were humanized and became drug candidates. And the antibody sonepcizumab to sphingosine 1-phosphate has completed two phase 1 trials and phase 1 and 2 phase 2 trials in renal cell carcinoma and age-related macular degeneration. Lapatamab is the antibody to lysophosphatidic acid, that other signaling lipid, has recently completed a phase 1 safety trial in healthy volunteers in preparation for a phase 2 clinical trial in neuropathic pain and traumatic brain injury. I'm not going to tell you about that work, okay? Because today is this, I want to talk about how this career and this development of antibodies against bioactive lipids, where it all began. It all began in the laboratory of Giovanni Salviati in 1990 with a very serendipitous, lucky discovery. And I'm going to go through that work. And I'm going to talk about the discovery of the role of sphingolipids in particular in muscle function. And that's what I want to talk about today. So I'm not going to tell you about the clinical development of drugs that resulted 20 years later from that work, okay? I'm going to talk about the first work that had to do with skeletal cardiac muscle calcium metabolism, okay? So this is the how it got started. This is Giovanni, of course, my mentor and my good friend. And this is a quote from Dante Alighieri. You can read the Italian, but you know, it's a very profound statement that I believe captures the sentiment of working with Giovanni Salviati, because we will never forget Giovanni Salviati, and he is like a signal of an ancient flame to us, to me in particular. So here's the first paper that we published. We published it with Romeo Beto, and actually Alessandra Teresi was a student in the Giovanni's laboratory. She's not here today, I don't think, but she was working with Giovanni, Romeo, and myself in this small laboratory, and we came up with this discovery. The discovery was that sphingolipids, in this case, one of the sphingolipids called sphingosine, not sphingosine 1-phosphate that you saw in the previous slide, but the precursor for sphingosine 1-phosphate is a molecule called sphingosine. It gets phosphorylated by a protein kinase, but the precursor sphingosine we discovered was involved in the calcium release from sarcoplasmic reticulum membranes of skeletal muscle. And here, we worked with the terminal cisternae membranes of sarcoplasmic reticulum. If you were to add those in the laboratory with a calcium-sensitive Indo-1 agent, you can make see the signal of the calcium flux into and outside of the sarcoplasmic reticulum vesicles, in particular the terminal cisternae elements of the sarcoplasmic reticulum vesicles. And if you add the terminal, you add calcium, you can see that the SR calcium pump will load the calcium inside the vesicles of the terminal cisternae, and then after you're finished loading these terminal cisternae vesicles with calcium, then you can induce a calcium release, either a spontaneous calcium release, or you can use a drug like a caffeine to open what we have subsequently come to understand is the ryanodine receptor calcium release channel of the terminal cisternae of the sarcoplasmic reticulum membrane. In this case, what we discovered is that the spontaneous calcium release could be completely blocked if you were to add sphingosine to the experiment. And then subsequent addition of ruthenium red had no effect. So we were pretty convinced that this effect of this sphingolipid was to block the ryanodine receptor calcium release channel of the terminal cisternae of the sarcoplasmic reticulum of skeletal muscle. And then if you were to do a dose response of sphingosine on either ryanodine binding in the open circles or calcium release. You can see that there's a relationship between the ability of the dose-dependent effect of the sphingosine in blocking the calcium release, coincident with its effect on preventing ryanodine binding to the ryanodine receptor. We did other work, so we said, okay, sphingosine that has this pharmacologic effect on the calcium release channel of the sarcoplasmic reticulum, but do the skeletal muscle and cardiac muscle have these molecules endogenous? Are they present in the muscle cell, in the cardiac and skeletal muscle cell? And the answer is a yes, because we published with Giovanni and with Romeo Beto and others that sphingosine is endogenous to both cardiac and skeletal muscle. And then we published with Daniela Beto and Romeo, we published kind of a review article indicating that the interaction between the dihydropyrin receptor and the ryanodine receptor, which, as you know, the dihydropyrin receptor of the T-tubule membrane interacts with the ryanodine receptor under normal circumstances to open the calcium release channel of the ryanodine receptor so that you get a muscle contraction, a skeletal muscle contraction or cardiac muscle contraction. And then we proposed that sphingosine had a binding site on the ryanodine receptor and could block that interaction between the between the dihydropyridine receptor and the ryanodine receptor and prevent that opening of the calcium release channel. We also did some other experiments in collaboration with Phil Palade, who at that time he was in Galveston, University of Galveston, and we wanted to look at this, see if there was any effect of these sphingolipids on other calcium channels. And a very important calcium channel, particularly in cardiac muscle, is the L-type calcium channel on the sarcolemma. What we did in collaboration with Beto and Phil Palade is do a patch clamp. And you can see that the calcium current of the patch clamp is here. But if you were to have inside the patch clamp this molecule sphingosine, you could completely block the opening, the calcium flux through the L-type calcium channel. And here is a control sphingolipid called sphingosine phosphorylcholine. Didn't have much effect. BSA, no effect. The role of sphingolipids then were extended through some collaboration with colleagues. I don't think Luciano Della Libre is here, or Giorgio Vescovo. Marco Sandri maybe is here today, I'm not sure. But we did some experiments to see whether or not there's other roles of these sphingolipids in muscle function. And in this experiment, we were studying something called cardiac cachexia. And you all know what cachexia is. This is when you have a heart failure. If you have heart failure, there's an interaction between the failure of the heart, particularly of a congestive heart failure, and skeletal muscle. There's skeletal muscle wasting and other effects that's called cachexia. And we did some experiments with with an experimental model of cachexia using a chemical called monocrotaline to induce the heart failure in the experimental animal. And then we discovered that the apoptosis in the skeletal muscle was correlated with blood levels of sphingosine and TNF-alpha produced from the heart. We also went on to do some subsequent work, not in collaboration with people here in the University of Padova, but other work showing that TNF-alpha acting through sphingolipids produced in the heart could be responsible for a negative inotropic effect of TNF-alpha. So heart failure associated with the overproduction of TNF-alpha could have some effect on skeletal muscle wasting, the cachexia response, but also a negative inotropic effect on the heart itself through the production of sphingolipids and the blockage of the L-type calcium channel of the cardiac sarcolemma. We looked at some other sphingolipids too. This is another experiment we did with Beto and Teresi. And we looked at the release of calcium from the sarcoplasmic reticulum membranes from the cardiac muscle induced by a sphingolipid called sphingosylphosphorylcholine. You can see that if you add sphingosylphosphorylcholine, there's a big inducement of calcium release and opening of a calcium channel. And actually, we discovered, subsequent to this paper, a new calcium channel associated with the SR of cardiac membranes. It was called scamper. And it was kind of an important discovery, because whenever you find a new calcium channel in the heart, and the importance of calcium in the heart is a very important function of the heart, to discover a new calcium channel that's modulated by sphingolipids, that was kind of an important discovery. And here shows you that you can take cardiac muscle cells, isolated ventricular myocytes. You can load them with calcium. They're beating, and the calcium flux is going up and down. There's calcium release, uptake, release, uptake, release, uptake. If you put them in calcium-free media so that you do not activate the surface membrane calcium channels, but were to add sphingosylphosphorylcholine, you could see that there's an inducement of the calcium release. Other experiments proved that that calcium, source of calcium, was the storage of calcium in the sarcoplasmic reticulum membrane. We went on to, in collaboration with Daniele Betto, pictured here. She's not here today because she's teaching here at the university still. We looked at the role of sphingolipids in muscle fatigue, and this now we're talking about sphingosine 1-phosphate, not sphingosine itself, but the phosphorylated form of the sphingolipid. And this was high-frequency muscle fatigue in extensor digitorum longus muscles, and you can see if you do high-frequency stimulation in the closed circles, over very few minutes you can get a loss of tetanic tension force, but if those skeletal muscles were bathed in the sphingolipid, the sphingosine 1-phosphate, there was a protection against fatigue, high-frequency fatigue. Dr. Daniele Betto went on to discover that sphingosine 1-phosphate is also protective in preventing a denervation atrophy in skeletal muscles. So you know about denervation atrophy. There's usually a reduction in 7 days after denervation. There's a reduction in the cross-sectional area of the muscle cell associated with the degenerative process associated with the denervation. But if you were to use, inject into the animal an antibody to sphingosine 1-phosphate, you can partially protect, or you could exacerbate actually the loss in cross-sectional area, indicating that sphingosine 1-phosphate that was endogenously present had a protective effect in the regenerating muscle. That if you were to block that sphingosine 1-phosphate with the antibody, then you make the atrophy worse, implying that sphingosine 1-phosphate had a protective role in promoting regeneration after denervation atrophy. Also, Daniele Betto did some other experiments promoting muscle degeneration with either injection, intramuscular injection of bupivacaine or notexin. In this experiment, in this publication, was use of bupivacaine to make a muscle degeneration, a muscle wasting, and then she looked at the response, the regenerative response as a consequence of the myotonic damage due to that bupivacaine administration. And she discovered that if you were to inject sphingosine 1-phosphate into the muscle at the same time you injected the damaging agent, the bupivacaine, you could protect that muscle from degeneration and actually promote a regenerative response. And similarly, if you were to use an antibody to sphingosine 1-phosphate, that promotion of the regenerative response is negated if you were to block the sphingosine 1-phosphate that's present endogenously in the muscle. She went on to discover the involvement of S1P2 receptors and S1P receptors in collaboration with other investigators at the university here, using either the knockout mouse model, knockout of S1P receptors, S1P2 receptors, or with some specific inhibitors of that receptor, you can invoke a role of S1P2 or S1P3 receptors in that regeneration response. And so all of that work and subsequent work with the role of sphingolipids in skeletal muscle calcium function, in cardiac muscle calcium flux, in muscle fatigue and muscle regeneration, and in cardiac cachexia, all led me eventually to work with the sphingolipids as targets of drug discovery. Along the way, I was pleased to work with some of these wonderful women scientists. Here is Sandra Furlan, who is at that time in the Margaret Lab, and we worked very closely together for many years. This is Alessandra Teresi, who actually did the first serendipitous observation of the role of that lucky discovery that the sphingolipid would block the calcium release channel. Here is Cristina Presotto, who is a graduate student in the lab. This is my lovely wife, Gail, who's not here with me today. Here is Daniele Betto. This is Doriana Sardona, who is sitting right here. And she looks younger now than she did when this picture is taken. But she's a very profoundly expert molecular biologist that worked with Romeo and others. And here is Gianna Facchetti Casano. And then along the way, I made many, many great friends, many of whom are sitting in the audience today. Here is Ernesto Damiani. I don't think he's here today, but I did many experiments with Ernesto. Here he is sitting with my lovely wife, Gail, and he became one of my best friends. Another one is Sergio Salvatore, which I see is sitting over there, and he's a great friend and collaborator, sitting next to the illustrious luminary Ugo Carraro, who is also a great friend. And I was fortunate to get a big hug from Paolo Bernardi, who is one of my very dear friends, and I'm glad to see him, and he has a beautiful family, and he's a very accomplished, excellent scientist. And then here also, is very happy to see that Tony Tolbadin is also present in the audience over here, and I worked with Tony Tolbadin and Sandra Furlan for many years in the Margaret Laboratory. And here is the famous friend, Romeo Betto, and what we are doing in his home is sharing the appreciation of Amarone wine and Carnia Stravecchio cheese from this area of Italy. And unfortunately, I was going to end the presentation with a video, but we had some technical difficulty. I was going to show this a few minutes of video of Giovanni when he was visiting my laboratory in San Diego State University. It would have been very beautiful to see actual motion picture video of Giovanni when he was a younger man and in full health. And I'm sorry I can't share that with you because we didn't have the right connector. But I'll end with this presentation with the quote from Dante Alighieri about the sentiment that was engendered by Giovanni Salviati and the Salviati family. So thank you for your very kind attention.